All right. Great. Thank you for the introduction, Guy. It's very nice to visit. I have known about neurophysiology in Israel for a very long time. I've been reading the papers since I was a graduate student in monkey work in general, and I know many of the people here that are doing this kind of work, so it's really fun to come and visit and see the labs. So as the guy mentioned, my lab is focused on trying to understand the neural systems in the macaque brain that underlie reinforcement learning. Reinforcement learning is a large area of study in computer science, but it's also basically a framework that we use to study learning in mammalian systems. And in mammalian systems, when we say reinforcement learning, what we're talking about is how we learn preferences from experience. Okay? So we make choices in life, and sometimes our choices lead to positive outcomes, sometimes they lead to negative outcomes. And if we're healthy, we want to learn from those outcomes to make better choices in the future, right? So obviously this is a very broad set of behaviors, covering things like learning which foods you like. So here's Andy Warhol about to enjoy a Burger King hamburger. They actually hired him to promote their hamburgers. I'm not sure if he's not looking like he's about to enjoy it. And here's a young lady who's obviously had a bad experience with broccoli in the past, or perhaps with something that smells like broccoli. So certainly learning which foods you like is a process of trying things and learning if it's something that you like. Now, when you push these systems too far, you can end up with clinical conditions, which is also a little bit of what I'm interested in. Obviously, drug addiction is a fairly severe clinical condition, and we can think about drug addiction as a compulsive approach behavior. And the definition of a compulsive behavior is a behavior that you continue despite intense negative feedback. Okay, so drug addicts carry on with their behaviors, their lives fall apart, but they continue to do this. So that's why we call this a compulsive behavior. We also could have compulsive avoidance behaviors, anxiety disorders. If you have agoraphobia, you may refuse to leave your home even though you lose your job and you lose relationships with other people. So there are aversive consequences to your behavior, but you don't learn from those consequences to adapt your behavior. Okay? So when these reinforcement learning systems are pushed too far, okay, you end up with clinical conditions. I've sort of set this up as a single axis, compulsive approach behavior, compulsive avoidance behavior. I would say these are probably not a single system. These are probably multiple systems in the brain. And I would say that we also probably don't know that much about particularly aversive behaviors, like which systems those really map onto. And I'm not going to talk about it today, but we are working a fair bit on that. All right, so that's, oh, and I should mention, any questions along the way, please stop and ask me. I'm happy to make sure things are clear. So we're all neuroscientists, I guess, or at least that's the goal of the work in my lab. So we want to understand the neural systems involved. And this is a coronal section through the front of the monkey brain. This is prefrontal cortex up here, principal sulcus, arcuate sulcus. And the striatum is thought to be a very important structure for reinforcement learning. And it's certainly the structure that I started working in when I started working on these problems. So I've outlined it here, and I've indicated the dorsal striatum and the ventral striatum. Based on the anatomical inputs in the front part of the brain, function varies more along this axis than it does along this axis, although there is some variation there as well. So that's why I tend to think of this as sort of a dorsal-ventral trade-off, right? But it's really which parts of prefrontal cortex are projecting into each part of the striatum that I think drives its function. So the striatum is important, and as I just mentioned, frontal striatal systems. So these are the anatomically defined circuits that connect prefrontal cortex into the striatum. So this is a lateral prefrontal cortex connecting to the ventral striatum, and there's an orbital frontal cortex circuit that projects into the ventral striatum here. Of course, dopamine is thought to be really important for these processes. Dopamine codes reward prediction errors during learning, and it's thought that the phasic changes in dopamine concentration in the striatum may be driving plasticity on these frontal striatal circuits here. And as a guy mentioned, I'm also going to suggest and show you data that supports the hypothesis that the amygdala plays a very important role in these processes. And the amygdala is normally not included in models of reinforcement learning, but the data we have suggests that it plays a fairly important role. So I'd like to start out by giving kind of the standard model to kind of put forward how I think a lot of people think about these systems. This is not my model. This has been developed over a number of decades by a bunch of different groups. And I will say that I call this the standard model, but one of my reviewers recently reminded me that this is a model and not necessarily the standard model, so I think it's a fairly well accepted model. This is going to be highly oversimplified, so if you are a basal ganglia aficionado, I'm going to leave out a lot of details, okay? So first of all, let's say you're in a supermarket and you want to choose between apples and oranges, right? So you go up to the bins and you see apples and oranges. Cortex represents your possible options and brings the information about the possible options into the striatum. So maybe a lateral prefrontal circuit connecting into the dorsal striatum here tells the striatum that you are currently choosing between apples and oranges. The striatum then selects an action based on value representations in the striatum. So to simplify things, we could say that maybe there's a population of neurons that represents apples and another population of neurons that represents oranges, and the activity level of those populations represents how much you value them. So if the apple population is more active, maybe you value apples more than oranges, and via some winner-take-all mechanism somewhere in the basal ganglia, you select an apple. So after you select an apple, you may take a bite out of it. Dopamine then codes whether that apple, the outcome, is better or worse than expected, right? So if the apple is better than expected, you have a positive reward prediction error because you predicted the apple to be pretty good, it was really good, better than expected, so you get a phasic increase in dopamine levels. And if the apple was worse than expected, you get a decrease in dopamine levels. And that change in dopamine concentration, that phasic change, drives plasticity on these frontal striatal synapses, right? So the apple representation coming into the striatum is strengthened if you have a positive reward prediction error, and it's weakened if you have a negative reward prediction error. So next time you go into the supermarket and you're confronted with apples and oranges, you'll have this updated representation of the value, okay? So that's how we compare apples and oranges, right? People often say it's hard to compare apples and oranges, but we learn from experience which of them we prefer. So these four steps can be captured by a fairly simple equation. This is really the Rescorla-Wagner equation, kind of written in a different way from 1972. And it's simple, but it describes behavioral learning pretty well, and it ties together neural activity in a bunch of different brain areas, right? So choice is represented in the cortex, values represented in the striatum, and reward prediction errors coded by dopamine neurons, right? So the idea is you have some baseline estimate of the value of your choice, and if you have a positive reward prediction error, you multiply that by a small learning rate, so you increase your value estimate the next time you encounter that choice. If you have a negative reward prediction error, you subtract a small amount from this value estimate, so the next time you're confronted with that choice, you have a slightly lower value estimate. So this is a pretty good equation. Sometimes I make a joke and say that the goal of my lab is to figure out what's wrong with this equation, right? Not that I think it's a bad equation. It predicts a lot of data and a lot of behavior, but it's an important problem, so we should continue to kind of tease it apart and understand the details of it. So one more specific model prediction here. There's a variant on the standard model called the actor-critic model. And in the actor-critic model, the ventral striatum is called the critic, and the dorsal striatum is called the actor, often. And the idea is that the ventral striatum represents values of choices independent of how choices are executed, right? So when you go to make a choice, whether it's via a specific action or picking a visual object someplace in space, the ventral striatum is going to code the values associated with those choices. Okay, that's a prediction of a particular interpretation of the actor-critic model that we're going to look at in a little while. All right. So my approach to this system is really kind of to think of it a little bit more in terms of not necessarily the computational principles that are often used to study it, but in terms of the anatomy and how information flows through different circuitry in the brain. And really what I do is I look at the anatomy and the kinds of information we've found in different circuits via neurophysiology, and I use that to try to understand what I think different bits of the striatum are coding. Okay? So we tend to look in prefrontal cortex and we tend to study eye movements. And we've done a number of experiments looking at the dorsal striatum's role in action outcome learning. And again, this is fairly specific, I think, to eye movements. So if you were learning reaching movements, different reaching movements that brought you a reward, you would be in the part of the striatal circuitry that's coding reaching movements, right? Which isn't really the caudate. The ventral striatum we think is important for stimulus outcome learning. And in the monkeys, we're always using visual stimuli. The amygdala we think is also important for stimulus outcome learning. And because now we have two nodes here in the circuit, both coding stimulus outcome learning, we think actually that the amygdala learns more rapidly when values change in the environment, and the ventral striatum learns more slowly. And the value of learning more slowly is that it makes you a little bit more immune to noise, which is often fast fluctuations in value when you're trying to make choices. So the amygdala can adapt more rapidly to fast changes in value in the environment, but it's also more susceptible to noise. Ventral striatum learns a bit more slowly, makes it a little bit more immune to noise. That's really kind of an operating hypothesis right now, but I'll show you a little bit of data consistent with that. All right, so first experiment. Any questions? We're good. So this experiment was done by my previous postdoc, Vincent Costa, who's moved on to his own lab right now at OHSU. In the first half of the talk, I'm going to be talking about lesion experiments. And in this experiment, we're going to compare 3 groups of animals. There's a group of control animals, unoperated control animals, and I think it's 5. There's 3 ventral striatum lesion animals. And we lesioned, if you do rat studies, you might think of differences between the core and the shell, but this is a complete ventral striatal lesion. Basically, everything below the bottom of the internal capsule here is taken out. So all this bit here. And we're going to compare those to... What's that? Yes, sorry, it's bilateral. Bilateral, yep. All these lesions are bilateral. Generally with unilateral lesions in these systems, you don't see any effects. So, and the other group is 4 animals that have lesions of the amygdala, and that's both the central nucleus and the basolateral and accessory basal nucleus. So basically complete amygdala lesions, complete ventral striatum lesions, control group, and we're going to see how they perform on a measure of reinforcement learning. So we use all choice tasks, and we tend to study these bandit tasks in the lab. These were used a lot in human functional imaging studies of reinforcement learning in the early 2000s. So we just ported those experiments into monkeys. So the way this task works, it's kind of a standard monkey task. Monkey starts out by fixating a central location on the screen. We present 2 images, left and right of fixation. The monkey makes an eye movement. So all of our choices are going to be indicated by eye movements. The monkey makes an eye movement to select one of the images, and then he's rewarded according to a stochastic schedule. Okay? So the way these schedules work is the task is executed always in 80 trial blocks. At the beginning of a block of trials, we introduce 2 new images that the monkey's not seen before. We assign to one of the images a high reward probability, the other image a low reward probability, and in different blocks of trials, we use different schedules, right? So if it's an 80-20 schedule, then maybe when you pick this image, you get rewarded 80% of the time, and when you pick this image, you get rewarded 20% of the time. If it's a 70-30 schedule, it will be 70% and 30%, etc. Okay? The schedules always remain fixed within a block. So during the acquisition phase, the monkey has to figure out by trial and error what the good image is, and the images are always being presented randomly left and right of fixation. So to figure out what the good image is, pick that as many times as possible, okay? Then on a randomly chosen trial between 30 and 50, it varies block to block, we reverse the choice outcome mapping, so the good image becomes the bad image and the bad image becomes the good image. The monkey has to detect that shift, switch to picking the other image, and finish the block. At the end of 80 trials, we introduce 2 new images. Monkey does another block, and we can get 20 to 25 blocks of trials out of the monkey a day. So a lot of learning. We're basically looking at this fairly rapid learning over and over and over again across blocks. Okay? Kind of experiments that are amenable to physiology studies. All right. So what do the lesions do to this kind of learning? So this is our 80 trial block. It's many, many 80 trial blocks, also averaged across all animals. This is the fraction of times the animals pick the best initial cue. So this is the cue that we assign the high reward probability to at the beginning. Black is our control animals. This is the 80-20 schedule. So we can see that the control animals fairly rapidly sort out the best stimulus, and then they pick it over 95% of the time. The dotted line is the reversal. This starts to come down before the reversal because we're smoothing a little bit in time here. Actually, it takes the animals about 1 to 2 trials after the reversal to pick that out in an 80-20 block and switch their preference. Okay? So the animals switch their preference and then they finish the block. Animals with ventral striatum lesions have a pretty clear deficit, and this is fairly well predicted by a large human imaging literature. But we find also that the amygdala animals have a substantial deficit. It's a bit larger than the striatal animals, pretty similar statistically, but a little bit larger. And this was actually fairly surprising to us, which I'll talk about a little bit more in a second. So this is the 70-30 schedule, and this is the 60-40 schedule. Basically, we see across schedules that the performance of all the groups gets worse because it's harder to figure out what the good stimulus is when the difference in reward probability is more similar than when it's more different. But the same rank order of performance holds across all these conditions. So we used a bunch of different schedules and reversals because there were experiments in the literature in humans using different schedules, reversals sometimes, reversals not. We wanted to put them all into one experiment so we could really look at it systematically. In most of the experiments we've done, we've not seen an effective schedule or something that's specific to reversals. So most of our learning effects that we find in these tasks are pretty consistent, independent of which phase of the task you look at or which schedule you look at. So there's an older literature in monkeys, started by Mort Mishkin, looking at what they called reversal learning. And these were behavioral tests where you had a Wisconsin general testing apparatus and there was a monkey in the box, and he would displace an object and there was a peanut under one object and no peanut under the other. And you would have always the peanut, for example, under the cube for a number of trials, and then you would reverse that. And in that literature and in those experiments, and in fact 2 of these animals, there are no deficits in that task. Okay? So when I started studying this, we designed this task completely differently because I have a physiology background and we try to control everything very carefully. But in some respects, right, you think, well, this is reversal learning, that's reversal learning, why aren't there any effects? And there are many differences between those experiments, and we haven't really tracked them down. But one difference that we did look at was we always use these stochastic outcomes, and in those old experiments, everything's deterministic, right? The peanut's always under the cube, or it's never under the cube. So we ran a deterministic version, and in this experiment, all the groups get better because it's easier. Obviously, one of the images is always rewarded, the other image is never rewarded. But we see, again, the same rank order performance, right? The amygdala lesion animals are still slowest, the VS animals are faster, and the control animals are fastest. So there's a lot of interesting sort of details buried in this, which I'll omit today, but you can go into the papers if you're interested in that. So point number 1 from the first study, and I will also mention that we actually did these experiments in the amygdala lesion animals by accident. So I had a graduate student in the lab, and as I was telling a guy, I asked her to do an experiment that was kind of tedious, and I said, after you do that, you can do whatever experiment you want. She said, I really want to study social processing in the amygdala. So I said, fine. She got some amygdala lesion animals from my colleague Betsy Murray and brought them in the lab. And we were just trying to train the animals on basic fixation learning stuff because we kind of run animals all through a standard training regimen. And they said, hey, the animals aren't learning to pick the right object. And so we decided to study it. But it was actually surprising to us on the basis of an older literature that the animals had a deficit in this. So really the take-home message from the first experiment, though, is that if you lesion the amygdala in a task that most people agree is a good reinforcement learning task, you see substantial behavioral deficits. And the second thing, which I mentioned a little bit earlier, is that the amygdala learns quickly and the striatum learns slowly. And the main data we have that's consistent with this, as well as a bunch of modeling, but you can just see it here. If we think that only the ventral striatum and only the amygdala are important for this kind of learning, or that they're sort of the dominant forces in this kind of learning, which may ignore an important contribution to the cortex that we haven't looked at yet, then when you lesion the amygdala, you're left with the striatum. So this red curve here is really the learning that the striatum can do without the amygdala. And the blue curve is the learning that the amygdala can do without the striatum, right? So the blue curve goes up much quicker than the red curve, which means that the learning rate is faster in the amygdala than it is in the striatum. So that's basically the evidence, along with a bunch of modeling and the other tasks that we have that's consistent with that claim.
Have you tried both of them?
We have not, no, but which is something we should do to see if you can completely, yeah, exactly. Yeah. It would be an interesting experiment to do. As you know, when you lesion the striatum, actually, these animals have a lot of behavioral difficulties, and we have to take care of them for a long time. So they recover actually quite well. But it would be you do challenging experiments like that all the time. So yeah. All right. So second experiment. So this experiment was done by a post-bac in the lab. So she just came out of her undergrad. It was not in grad school. Katie Rothenhuffer. She's now working for Bill Stauffer now, actually, who worked with Wolfram Schultz, recording from dopamine neurons. But she was extremely productive. She did this experiment in 2 years, which is tough. So now we're just going to look at the ventral striatum lesion animals. We're running the same paradigm in the amygdala lesion animals right now, but we don't have the data yet. So we did the VS, and we published it separately. So now we're going to use a slightly more complicated task. Basically, it has 2 conditions instead of 1. The basic task design is the same. So 80 trial blocks. At the beginning of the block, we introduce 2 new images. The animal has to learn and reverse, just like the other one, reverse somewhere in the middle. But now we're going to assign the reward probabilities using 2 different conditions that are fixed for a block. So if it's the what condition, it's going to be exactly like the first experiment. What we're going to do is we're going to assign a high reward probability to one of the images, a low reward probability to the other image. The monkey has to learn the good image, reverse in the middle, etc. If it's a where condition, we're going to assign a high reward probability to one of the locations, a low reward probability to the other location, and the animal is going to get rewarded for making a rightward saccade, rewarded more frequently for a rightward saccade independent of the image located there. Okay? So we don't cue the condition. The animal has to learn by trial and error at the beginning of the block whether it's a where or a what block. And the blocks stay fixed for the entire 80 trials. And then after 80, we introduce a new block, which could be either, etc. So the animal has to learn the condition, and then within the condition, learn either the object or the direction. And we run these in randomly interleaved blocks. So here's the performance again. This is our 80 trial block. I'm plotting this in the same way. This is choice of the best, in this case, initial Q, because this is just the data from the what condition in the 80-20. Here's the control animals. They don't get quite as high because it's a harder, they have to infer the condition as well as what they should pick. And here's the VS lesion animals. So this is actually the largest deficit that we saw in the VS lesion animals. And this experiment was run after we ran the previous experiment, and these are actually the same 3 animals. So they had 4 or 5 months of experience with just doing what before we started doing this task, and their performance actually dropped in this experiment relative to what they did in the previous experiment. Okay? Now if we look at their performance in the where condition, it's actually rock solid. So they're actually slightly better via some analyses at learning to pick one of the 2 directions than the control animals. And basically what happens is when we introduced the where condition in this task, the animals tried very hard to use the where strategy in every block of trials, and only after many, many, many failures would they actually switch to trying to figure out which of the 2 images to pick. So that's why their performance actually dropped here. And if you look, you can see averaged across blocks that they're perseverating with the where strategy even in the what blocks. Okay? So the ventral striatum does not play a role in action outcome learning. Again, this is specific to eye movements. We think there probably are a lot of differences between the eye movement system and the reach system in terms of learning. We haven't started studying that systematically yet. There's a few things around that make that suggestion. So I'm really qualifying everything and saying that this is an action, but it's an eye movement. The ventral striatum is not the critic. So if we go back to the introduction, I introduced the actor-critic architecture, which suggested that the ventral striatum should code action values independently of how you make your choice. And here we say when the choice is an action, you have no deficits, and when the choice is figuring out which of 2 images to pick, you do have a deficit. Also, we did see that the animals with ventral striatum lesions got even worse in the what condition. We know they can do better than they were doing. So it's possible that animals with ventral striatum lesions are having a hard time switching from action outcome learning to stimulus outcome learning. It's also true, as if any of you here work with monkeys, that if you interleave hard and easy conditions in a task, they will just kind of cheat through the hard conditions to get to the easy ones and do those. So I think our animals were just not trying as hard as they could have, whatever that means. But all right, so everything I've told you up until now is published, and we sort of consider this our sort of outline of how we think the subcortical circuitry for reinforcement learning is organized. You know, we've been working on this for 12 years or something now already, 11 years, a long time, longer if you consider when we started these experiments. And just to give you the outline that I gave at the beginning, the dorsal striatum underlies action outcome learning. Ventral striatum is involved in visual stimulus outcome learning. The amygdala also does visual stimulus outcome learning, and the striatum learns slow and the amygdala learns fast. And again, I'll just emphasize that our ideas about what kind of learning is going on in different subparts of the striatum really follow from the input that each part of the striatum gets from prefrontal cortex and the information that's being encoded in that part of prefrontal cortex. And I think also by starting at the poles of this system, we've got our best chance of finding differences because there's really kind of a gradient of function as you go across the striatum here. And the amygdala is really a very sensory structure. It's not very close to motor outputs, so it gets huge visual inputs. So it kind of makes sense that it's actually having a role in this kind of visual stimulus outcome learning. All right. So now we're going to move on from our subcortical circuitry to some recordings we did in dorsal lateral prefrontal cortex. Again, I kind of divide this circuitry into a dorsal circuitry and a ventral circuitry based on the cortical location of the input to the striatal location. So I had the red and the blue circuits. This is the blue dorsal circuit. And we're going to focus on dorsal lateral prefrontal cortex primarily because we had an opportunity to do some high channel count recording studies there. So working with a very good surgeon, Richard Saunders, Richard implanted 8 Utah arrays in dorsal lateral prefrontal cortex. So he put 4 in the left hemisphere and 4 in the right hemisphere. Each array is 96 electrodes, so I think that's 768 electrodes. Ramon ran the experiment, collected the data, and then did a lot of spike sorting. And we ended up with, currently we have more sorted than this, but the data I'm going to show you is from 6 sessions, 3 from each monkey. We got 585, 747, 677, 1,026, 877, and 598 neurons in each individual session. So some really very large ensembles recorded simultaneously. And spike sorting is a really annoying thing to do, so Ramon has a very serious look on his face. He developed that during spike sorting. And I would go into his office and say, Ramon, have you sorted the spikes yet? Eventually he learned to give me this fairly aversive look. And, which is basically like, leave me alone, I'm working on it. Yeah, so now we're going to move into some slightly more complex analyses. And I'm going to try to unpack these clearly for you. But the basic idea that I'm going to suggest, so a lot of people are really interested in recording from large populations of neurons simultaneously right now, and a lot of people are interested in studying the dimensionality of the representation in those. And approximately the dimensionality is, if you know about principal components analysis, right, it's how many principal components do I need to account for some large fraction of the variance in my data. And there's the idea out there that we generally don't need that many principal components, even if we go up to 1,000 neurons or something, okay? And there's some fairly nice papers now describing why we actually expect to see that. Right now, I think they're still on bioRxiv, but there are some nice theory papers around. But we're going to go after that question in this data set. We're going to use it specifically to address some behavioral questions that have to do with reinforcement learning. So the questions we want to address is, how does the brain maintain expected reward representations across the many choices with which we are faced, right? So in every block of trials, the monkey's getting new stimuli, and he has to learn to associate values with 2 new stimuli in every block, and somehow he has to keep those separate from the stimulus outcome associations that he had in previous blocks. So how does the brain roughly solve that problem? And furthermore, what happens when we reverse the stimulus outcome mapping in the middle of the block here now, right? So now we have the same 2 stimuli or the same 2 actions, but we've reversed the values associated with them. What happens to the patterns of neural activity in prefrontal cortex in these large populations? And just as kind of a straw hypothesis, and I'll just tell you now, this is not what happens, we could say, does prefrontal cortex use a common coding space to represent choices across different options? In other words, does prefrontal cortex just say, I'm now picking a good stimulus that I know is a good stimulus, or I'm now picking a relatively bad stimulus, and those should diverge with learning. So a lot of models in orbital frontal cortex actually make the assumption that OFC neurons just code the value of a choice, and they don't say anything about whether or not that value coding is associated somehow with a specific visual stimulus. Actually, in all of our data, we have a lot more physiology data than this. We never see that. So it's always the case that value coding is specific to stimuli and it's not independent of the stimuli. So the task we're going to use is the same what versus where task that I was just talking about. This is just the behavior, so exactly the same behavioral plot that I've been showing you, just showing you that there's good learning here. These are the 2 animals used in these studies, and this is the behavioral data from these 6 sessions. So this is the what condition, this is the where condition. These 2 animals were a little bit better at the what condition, at least in these sessions. I should say most of the behavioral data that I showed you, we get like 30 sessions from each animal, so we really collect a lot of data to get nice modeling fits and all this stuff. So this is a much smaller sample of behavior. All right, so how are we going to analyze this data and address our questions? So we've got about 750 neurons recorded simultaneously. The first thing we're going to do is throw out the early learning trials, right? So we're going to throw out these first 4 trials in every block before the learning approximately asymptotes. Our answer doesn't depend specifically on this, but we're just trying to get rid of this early activity and look at just the activity when the animal's picking the option they've learned is better. But remember, even when they've more or less learned it's better, they still make a fair number of mistakes. So we get a number of trials associated with each of the choices, even after throwing out those first options, okay? So now we take the rest of the data during acquisition here, and we sort it into the trials where they chose object 1 and the trials where they chose object 2. And if we just had 2 neurons recorded simultaneously, neuron 1 and neuron 2, we could look at the average activity when they chose object 1 and the average activity when they chose object 2, and the circle would be sort of the one standard deviation or the distribution of responses. And we could fit a discriminant line by doing a kind of linear decoding analysis. And really what we're trying to do is we're trying to find one dimension out of the 750 dimensions, which when we project the activity on that one dimension, gives us a good separation of these scalar distributions of activity that allow us to predict which choice the animal made. Okay? So we're doing linear decoding, and we're going to get that line out that actually we're using for doing the decoding, and we're going to look at how that line changes across blocks. Okay? So we fit a linear decoding line to the acquisition phase. We fit a different one to the reversal phase. We do that in every block of trials. We have 24 blocks of trials, 12 of each. We also use 2 time bins, but you can ignore all those details except for the fact that just tells you that we're going to get 96 of these W's out of the data per recording session. Not really important, but you're going to see that our axes go to 96. Okay? But basically what we want to do is we want to look at how much these W's vary across blocks of trials. And W is a measure of the population code for learned value in DLPFC. And because we're looking at it for every different acquisition and reversal phase in every different block, we can see if W varies as you learn different images. And this gives us a measure of whether or not the population code for value depends on the image that you're learning the value about. Okay? So the way we can do that is we can take all of our W's from the different blocks. K goes to 96. We can do singular value decomposition or principal components analysis on W. Look at whether or not we can span the W's from different blocks using some low dimensional basis set, right? So if I can use a few dimensions to span the W's from all my blocks, that would mean that prefrontal cortex was just reusing the same representation every time we learned a new pair of objects, right? But if the W's vary a lot across blocks, then I'm going to need more dimensions to account for them. And that would mean that neural activity was varying, was sort of sitting in different parts of population coding space every time we learned a new pair of objects. And we can look at the same thing for directions. All right, so if we do this across the whole task, lumping everything together, this is what we get. And we see that we need about 25 dimensions to account for 80% of the variance, 50 dimensions to account for 90% of the variance. But by itself, that really doesn't tell us very much. We could compare to other tasks if we want. Interesting, a lot of people think that you need between 5 and 10 dimensions, so we're seeing more dimensions here. But you know, we're analyzing our data a little bit differently. There are a lot of differences. So what we really want to focus on is differences between our conditions, and then ask whether or not we can understand those differences. Okay? So this sort of busy plot here, the orange line, so orange-red line is a random basis. This is the unity line. But the two that are most important here are the where condition. So this is when the animals are learning directions in green, and the what condition when they're learning objects in blue. And basically, to account for 80% of the variance, you need about 14 dimensions in the where condition and about 20 dimensions in the what condition. So activity is higher dimensional across blocks of the what condition than it is across blocks of the where condition, right? Task dimensionality is higher in what blocks than where blocks. That means that these W's, the population code for value, are more different across what blocks than across where blocks, okay? So now what we can do is look at that same thing another way, right? So we have these W's that we've picked for each of our individual acquisition and reversal phases. We can project the neural activity onto those W's for a particular phase, and we can measure the separation between these distributions using D prime, which is just kind of a signal to noise ratio. And the higher D prime, the better we can do decoding. But D prime is a continuous metric, right? It's not like 87% correct, it gives us a continuous number, all right? Furthermore, what I can do is I can take the W from the reversal phase and I can project the acquisition data onto that W, right? Or I could grab a W from a different block and project the activity on it. And after doing that projection on different Ws, I can compare how well separated these distributions are, okay? So in other words, I can look at the projection of my choices here, of the population representation of my choices, onto either the W for this or the W from reversal. And the hypothesis is that when I use this one, I'm going to lose information because this one is going to be somewhat orthogonal to this one. It's going to point in a different direction in population coding space, right? So if we think of this in a low-dimensional space, basically if this is the projection of my activity on the W for just acquisition, right? If I grab an orthogonal direction, W2, and project this activity on it, it's all going to actually project into a point down here, and I'm not going to have any separation. So measuring the separation in the distributions tells me how orthogonal my W's are from different phases, okay? And this is an equation that captures that relationship, which is really the same question I was asking when I asked how many dimensions do I need to span the W's from all the different blocks, right? Because if the W's are very different, right, to span W1 and W2 as I've drawn it here, I need 2 dimensions. Whereas if they point in the same direction, I just need 1 dimension. And furthermore, if they point in the same direction when I project the neural activity onto them, I should preserve information. Which is a mouthful, I'm sorry, but we're trying to deal with this high-dimensional data, and this is our approach to doing it. All right, so let's see what happens when we do this projection and compare between the what and the where conditions. So what we find is this is the projection, and this is all cross-validated as well. This is the projection onto W. This is the information about choices when I use W from the current block. This is when I use the reversal phase, or if I take the reversal phase data and project it onto the acquisition phase. This is a different block of the what condition, and this is using a W from the where condition. So the point here is that I lose a lot of information when I use a W from a different block, which means that the W's point in a very different direction across different blocks of trials, okay? Now if I do the same thing for the where condition, I lose a fair bit of information when I use the opposite phase, but if I grab a W from a different block, it's actually pretty similar, okay? Which is consistent with probably an intuition that, so the directions are the same in every block of trials. The animal is just to figure out if it's right and then left. And if direction coding is fairly preserved across blocks, then I expect to get this, right? I might have a little drop due to some non-stationarities or something, but as long as my direction coding space is maintained, then I expect this to be roughly true, okay? So basically, this dimensionality that I see here when I look across all the blocks, we can measure when we compare single blocks, right? Because the drop in information when I use a W from a different block means that the W's from these other blocks point in very different directions. So if I take a bunch of these and I try to span them with a single basis, I'm going to need more of them, right? So the data for the what condition is higher dimensional, okay? I can look at that one more way, which is to actually just measure the angles between these. And if you have, you can measure something called the principal angle between two different two-dimensional bases in high dimensions, and that's what this is. So if I look at the angle between my informative dimensions in the what condition, right, it's about 80 degrees. And because of stochasticity in the data, even if they were perfectly orthogonal, I would only expect them to be about 86 degrees different, right? So these guys are quite different. They point in very different directions. If I do the same thing for the where condition, I'm closer to about 70 degrees. So there's more similarity in the directions for the where condition than there are for the what condition. So again, yes?
You try separating the what condition into the different directions, because still you have what to the right and the same to the left. Maybe Cortex just codes for where, also in the what.
It does, actually. So in other words, if you look at whether or not you can recover direction coding in the what condition, you can. I'm not showing that here, but it works very similarly to what we're seeing in the where condition.
But wouldn't that explain this?
Well, no, that's kind of exactly what I'm saying, is that there's preserved coding. So wouldn't that explain this versus this, you mean? Yes. There's preserved coding of direction across blocks, right? But because the visual images vary, every time I present a new pair, they get represented in a new sort of direction in prefrontal cortex. Yep. That's 3 different analyses here, all more or less showing that same thing.
So,
Yeah.
same as saying that in the wear condition, they're just encoding less the visual stimulus.
Yes, exactly, right. It's actually in the data, but I can linearly throw that out, right? So if you look at single neurons and look at an ANOVA and put visual stimulus in as a factor, it's a huge factor, even in the wear condition. It shows up later, things that you would expect, but you can linearly decode that and throw it out. Exactly, which is what we see here. Yeah? Yes. You mean, so basically, like in the wear, when I'm saying cross block here, that's taking like block 1 versus block 7, for example. In block 1 versus block 11, I do all pairs and then I average all pairs. Okay? Let's see. Okay, so the only last point that I want to make is it's not just a representation in prefrontal cortex of the visual image or of the action. There's actually learning on top of that, okay? So the information about the values of the 2 chosen images doesn't show up right away. Remember, in these analyses I've been showing you so far, I threw out the first 4 trials and just looked at the activity after it had somewhat stabilized. Now we're going to look at those first 4 trials and see how those representations develop, how that sort of dimensionality develops. And you could do the same thing after reversal, but I took those slides out in the interest of time. So this is 2 dimensions of neural activity. So I projected my high dimensional activity down on 2 dimensions and I'm just plotting it. This is the first trial where the animal chose object 1, and this is the first trial where the animal chose object 2. And then these lines connect the subsequent trials where they pick each of these objects. And the circles represent what I would call the learned distribution. So that's the distribution of responses from the data after I throw out the first 4 trials. Okay? So what we see is that there's a lot of overlap in these representations early. FT2 is basically sitting right on top of FT1, which then diverges later, okay, to these distributions which are better separated. Okay? So I can take each individual trial here and I can measure its distance to this distribution, and I can take these trials and measure their distance to this distribution and compare those, and I should see that that the information about the options is basically showing up as the animal's learning. Okay? That's for the what condition. This is for the where condition. We see a little bit of an effect here, so these guys are kind of hanging out in the middle here, but then they quickly converge to these stationary distributions over here. Okay? So if we just average that across all of our data sets, we can basically watch these distributions kind of settle in across space. So this is trials at the beginning of the block, starting from trial 1 going out to trial 25 here. This is the difference in information, comparing single trials to their correct distribution and incorrect distribution, so this should go up if they're learning. We see that early on, there's basically no difference in the information for the 2 choices, and then as they learn, the information about the 2 choices grows. Okay? If we do the same thing for the where condition, we see that early on there is some separation, so there's information about the direction of the saccade early, and this is somewhat a consequence of the fact that there's this preserved direction coding, right? But even in the where condition, this grows somewhat before it stabilizes later. Okay? So, conclusions. So basically, just from this last part of the data here, from the physiology experiment. So how does the brain maintain expected reward representations across the many choices with which we're faced? Basically, for visual images, it uses different locations in coding space in prefrontal cortex to represent the values for different pairs of objects. So it can easily keep these straight because they sit in very different regions in coding space. How does it change when the values of choices, when they're just reversed? So I didn't go into this in a lot of detail, but even just reversing the choice outcome mapping changes by quite a bit, actually, the region of space that this information is encoded in. And we saw that actually when we compared information in the acquisition versus the reversal phase, or the reversal versus the acquisition phase. There was a big drop in information when I used the W from the other phase. And this is true even though it's actually the same actions in those other phases and the same objects being picked. I've just changed whether or not they're going to get a juice reward later when they make a particular choice. So prefrontal cortex doesn't use a generic coding space to represent choices of objects, but because my two directions are the same, it actually does reuse that space to a large extent for coding directions. And then I say because learning, what did I say here? Over a new pair of objects or reversing their values drives activity in a new part of coding space, it increases the accumulated dimensionality of DLPFC representations. So I sort of mentioned this earlier on, but basically a lot of people are looking at this dimensionality in fixed tasks. So a lot of people in the reach system are interested, in the dimensionality of motor cortex representations in the reach system, but they use a small set of reaches, and so you end up with basically a relatively low dimensional representation. In our task, because we have the animals relearning new objects all the time, we actually see that the dimensionality grows every time they learn a new pair of objects. You can also compute these curves actually by accumulating across blocks. Basically, I compute the dimensionality of 1 block, of 2 blocks, of 3 blocks, etc. And it grows also as you look at change in dimensionality across blocks. So, all right, and so I usually just close with this as a way to say that studying these systems that have this very looped architecture is fairly complicated. I would say that we don't have good models of how to study a system this complicated. We're using lesions and physiology, but lesions make a lot of sense if you're in a system where you can basically stop the flow of information. So if you lesion the LGN, you go blind, right? But if we look at a system like this, lesioning any node in this system really can't stop the flow of information because there's another route for that information to get there. There is behavioral evidence, some from our lab and some from other labs, that lesioning any node in this system actually affects behavior, but to be honest, we don't really know why. We can lesion all those nodes and affect behavior. The effects on behavior that we look at are always like, well, they don't do quite as well. They learn more poorly, right? And we have a little bit of hypothesis that the amygdala might be learning more quickly than the ventral striatum. But I often encourage people when I give talks like this to start thinking about how we can study systems that are this complex, and give us people some ideas to help us move forward. Anyway, that's the lab. So I think I pointed out everybody that did the work. So this is Ramon who did the physiology. Vinny did a bunch of the lesion work. And Katie also did one of the lesion experiments. And one more point. So the lesion monkeys I actually get from my colleague, Betsy Murray. Those lesion surgeries take a long time, so I have to give her a lot of credit for doing those. They're very hard. The array surgery was done by Richard Saunders and Phil Browning. And all my funding comes from the NIMH intramural program. That's it. Thank you.